Hi Jobbies. So today we'll have our introductory lesson in biology. We're looking at the cell cycle. So in this lesson we're going to have an overview of the cell cycle and then we go to interphase and we look at the phases under interphase. So this will be a very short topic. So the lessons will be 3 under this topic and we'll be done with the cell cycle. We'll keep them very short. So we can start with the overview. So firstly, to introduce this cell cycle. What is the cell cycle? So the cell cycle, it is simply the sequence of growth, DNA replication, growth, and cell division. So that is the basic idea about the cell cycle. It involves these 4 stages. So a cell will usually be small, and then in its smallness, the DNA inside it will replicate. That is DNA replication. So like if we have 23, the DNA will be 46. If we have 10, it would be 20. Right. So the cell is small. It will start growing. As it grows, and then it will undergo DNA replication. So the next cell should be at least bigger, so that there's at least enough room. And then after DNA replication, there will be further growth. So the cell will grow again, and then after growing further, the cell will now undergo division. So it will undergo cell division, so that we can have 2 daughter cells. From these cells. So this is a cycle. So remember, this is a cycle. So it continues, it goes on and on. So even the 2 daughter cells, they can grow, and then later on they also replicate, meaning they'll form other daughter cells. Even here, other daughter cells, they'll grow, they'll replicate. The cycle will continue just like that. So the cell cycle is important for tissue repair. In case cells in the body are damaged, the cell cycle will be there to ensure that the cells are regenerated. Alright, so that's the introduction to the cell cycle. So now let's have an overview of this cell cycle. So we're going to have this is the general cell cycle. So that's very, very bad. I hope you read it better. So I like summarizing biology using organograms, this which is also called the organizational chart. So the cell cycle, what do we find in the cell cycle? There are 2 phases in the cell cycle. So these phases, we have interphase here. So we have interphase. So there is our interphase. So one phase is interphase, and then the other phase it's mitotic phase. So if you have space, you finish it up. Mitotic phase. So these are the 2 phases of the cell cycle: interphase and mitotic phase. So under this lesson, we're going to focus on this. and then in the next lesson we'll focus on mitosis, and then in the last lesson we'll deal with meiosis. So interphase, what does interphase involve? So these are the two branches of the cell cycle. So interphase, it has okay, so it has the first gap phase. So we have a G1 phase. So this is a G1 phase, and then we have an S phase and a G2 phase. So you can also think of these phases as from the definition itself. So what did we define the cell cycle? We said the cell cycle, it is the sequence of you can see here we have growth, the sequence of growth, DNA replication, again growth. So this is G, replication, simply DNA synthesis, that is the S, and we have another growth. Yeah, so you can think of it in that sense as well, if it would help in remembering. So these are the three phases under interphase: G1 phase, DNA replication phase, G2 phase. Yeah, that's all. And then now moving on to the mitotic phase. So under the mitotic phase, we have two phases. So we have karyokinesis and cytokinesis. Okay, so these two, karyokinesis and cytokinesis, these are the two processes of mitosis. So karyokinesis, this will be our next lesson. Yeah, so under karyokinesis we have four phases. So we have prophase. So this will be our prophase. So we have prophase, and then we have metaphase. So after prophase we have metaphase, there you can finish it up, and then after metaphase we have anaphase. So we have anaphase. So this is metaphase. Okay, and then the last one is telophase. Telophase. That's the last phase. Alright, and then under cytokinesis we don't have something significant to list, except the cleavage and furrow. We'll look at all those. Alright, so this is the skeleton of the cell cycle. So the cell cycle has an interphase and a mitotic phase. Okay, and then under the interphase we have G1, S, and G2. Under mitotic we have karyokinesis and cytokinesis, and the other name for karyokinesis is nuclear division, and for this one it's cytoplasmic division. So cytokinesis is whereby that part where we said the nucleus will now, the cell will divide. So for the cell to divide is the last stage, it's cytokinesis. Alright, so the way I was writing them, that's how they follow each other. Like it starts with interphase G1, G2, and then we go to karyokinesis and then cytokinesis. Okay, so having drawn our overview, we'll then go on to focus on this. So this chart, we can write it in form of a circle. Okay. So one thing you should remember is we have 4 things. So this is like the entire cell cycle. So in the cell cycle, we have G1, G2. So first let me do this. Okay. So I was saying, in the cell cycle, we have gap 1 phase. So I like calling it growth phase. Yeah, but it's gap 1. So we have the first gap, and then we have S phase, which is DNA synthesis, and then we are going to have gap 2, and then the last one, it's mitosis. Okay, so this is from the definition. We said it is growth, DNA replication, growth again, and cell division, which is mitosis. So one thing you should always remember is this part from here, the whole lot of this G1, S, all the way up to G2, this part is interphase. So this is interphase. And then this part which is remaining here for M, it is mitotic phase. So meaning the prophase which we are writing, they are inside here of prophase, metaphase, anaphase, and telophase. Yeah, and I'll tell you about the mnemonic I made. Yeah, so that will be for when we come to this. Yeah, so interphase, it takes up a lot of time. Like it takes 6 hours for a cell cycle to reach up to G2 phase, gap 2. Yeah, and interphase is the longest. It is longer than mitotic. I think you can see just from here. Yeah. Okay, so I think we've explained on that. Yeah, so just remember the whole lot of this part, this is interphase, and that is mitotic. Okay, so we want to discuss interphase in this lesson. So that is so we just use the same diagram. Okay, so interphase, it is where remember a cell is basically small. It will have to grow. This is G1, and then when it grows, DNA will be doubled. So like in G1 we could have maybe one chromosome, but under S phase we're going to have 2. This means that DNA replication has taken place. So this is the S phase, and then after the S phase, the cell will grow even more. That is now G2 phase. Are you following? Yeah, so under G2 phase, meaning the DNA is replicated. Like from here we have 2 there. Yeah, so we just want to explain these same processes. Yeah, so like what happens and pretty much everything else necessary. Okay, so we start with G1 phase. So remember, for a cell to be able to successfully replicate, for a cell, yeah, for a cell to successfully replicate, at first it will need to grow. Yeah, so at the G1 phase, that is where the cell will grow. So the cell will grow there. So it will keep growing at this phase So like as time goes by, I think I mentioned like it takes about approximately 6 hours for interphase to take place up to this point. So the cell, the interphase will start proceeding. Yeah, so the cell is growing, it's growing, it's growing. So as the cell is growing up to, and when the cell is growing up to a suitable size, or when the cell is now like ready to go into the next phase, there will be what is called a checkpoint. Okay, so we have a checkpoint. So we have a checkpoint. So a checkpoint is where now the cell, it will have to like hold the processing, like it will take a short break, like hey, is everything okay here? G1 wants to change to S, so can we allow it to proceed? So at the checkpoint, this is where now the cell, it will have to check like if all is well. So, and in the G1 phase, so this is gap phase, but like I said, I like mentioning it as growth phase. Yeah, so in this phase, this is where we the cell organelles. Yeah, so the cell organelles like mitochondria, the nucleus, at this point, this is where they start like becoming bigger. Yeah, they start becoming larger in size. That is on the G1 phase. So they'll reach a point before going to the next step, they'll reach the checkpoint. So what is the purpose of this checkpoint? What does it do? Yeah, so let's quickly have a look at that. So we have here, so this is at G1 checkpoint. So the cell is checked if the cell is large enough, if the environment is suitable for the next phase. Yeah, so those are the major things which take place at this checkpoint. Yeah, the cell will be checked if it is large enough and if the environment is suitable. So you would want to ask, so what if the cell is not large enough? Like instead of the cell being at this size, it's looking something like this. If it happens like this, there is what is called a cell cycle arrest. Yeah, so we have a cell cycle. So cell cycle arrest. So this is where the process is stopped. Like the cell, it won't undergo the S phase because it doesn't meet the requirement. Or in some cases, if the cell goes through like if the cell is small but it starts undergoing the S phase, it means that there will be need for more protein synthesis, so that at least the cell is made to become something like bigger, yeah, and all that. So this is what happens at G1 phase. Yeah, it simply just grows and there's a checkpoint. In the checkpoint, it will check if it's large enough, if the environment is suitable for the next phase. So if the cell at this point is like having what can we say, if we can call it like pathogens or things which will make replication not to take place, then the cell won't go to the next phase. Yeah, so we just take the basics and yeah, we proceed. Yeah, so this is it on G1. So now we go to the synthesis phase. So what is this synthesis phase. So this is DNA replication, and this is whereby, and the other thing on the G1 checkpoint, it checks if the cell is ready to undergo DNA replication. So in the G1, in the first phase, like I think I said, if we have like one chromosome, it means here, and after replication takes place, we would have 2, because replication has taken place. If this particular cell has like 20, then in this there will be 40. Yeah, and it will proceed. So we went so in the S phase it's very fast, like there isn't much, but we looked at DNA replication. Yeah, all those processes, those enzymes, they take part here. Yeah, you have to remember the polymerase, all those, the double helix. Alright, so after the S phase, so at the S phase there is no checkpoint. Yeah, so this is just passing for free. Okay, and then from this point the cell continues with the journey. So at this point now, the cell has replicated. So what is remaining is for the cell to become even bigger, so that it's ready for mitosis. So at the G2 checkpoint, so here at this G2 checkpoint, we want the cell to be bigger. So it was like this, but want something even larger. So what happens at the checkpoint, it's quite similar to G1, but here at the G2 checkpoint, it is basically where the cell is checked again. So like before the cell starts to undergo mitosis, it will have to be checked, like okay, replication has taken place, but is the cell completely, are the cells replicated nicely? Like, is there any damage? Is there all such thing? So all such things, they'll be checked again by G2. So at the G2 checkpoint, so before mitosis again, there will be another gate. So we're going to have a police here. If it is found that the cell is not fully replicated, the cell, it won't go to the next phase. Yeah, so this is like this. We're looking at this cell, like we started with this cell here. It had one. It was small at first, and then it became bigger, and then it had one chromosome, and then the chromosome replicated to 2. From there, after replication here, it has increased. The cell is now bigger, and then now since it's now bigger, then it can now go to the other phase. But before it can go to the other phase, it should be checked. Did everything work just very well? Is the cell large enough for replication? Because remember, mitosis, it's this. Under mitosis later on, it is where the cell in the end should be able to divide. So if we have a small cell, replication won't be very easy. So no wonder the cell should be big. So but even if the cell is big, like this, are the things inside okay? So that is the purpose of G2. It checks if replication took place, it checks if the cell is large enough, and then it also checks if the environment is suitable for the next phase. Alright, so that's on the G2 checkpoint. So we have these. So I think I've talked about them. We have to check if DNA has replicated, to check if the cell is large enough, to check if the environment is suitable for the next phase. Alright, so this is the this is interphase, G1, S, and G2. So when we go to M phase, that's mitosis. That's mitotic phase. So actually this one it's the longest, but in explaining it's even the shortest. Yeah, so mitosis, we'll look at it in a later lesson, in the next lesson. So under mitosis, I'll just introduce and leave you with a mnemonic as we proceed. So when we come and look at mitosis, so mitosis is simply a point. So this is under karyokinesis. So this is where the contents in the DNA, they are replicated. Okay, so and then we said karyokinesis has 4 things. So this is karyokinesis. So pardon my handwriting. I'm taking the excuse that no, I'm a medical student, so I'm writing badly, but I just have a bad handwriting. We shouldn't lie for doctors. Okay, so that was a short break. So under karyokinesis, so this is karyokinesis. And for those of you who want to do medicine, the good part is that if you don't know the spelling, just write karyokinesis. Stress free. Okay, so under karyokinesis, it is under mitosis. We have 4 processes which take place. So the 4 processes, we have prophase. Okay, so we have prophase, and then we have metaphase, and we have anaphase. Prophase, metaphase, anaphase, and telophase. So these are what we have. Writing is hard. Okay, so we have prophase. You can give up in 2025. Prophase, we have, this is metaphase. Yeah, so you'll see them nicely in lesson 2. Metaphase, we have anaphase. We have anaphase. I'm looking at the handwriting, I'm like, wow. Okay, so we have anaphase and we have telophase. So these are what we'll come and look at. Just these, and that will be our lesson 2. So sometimes you can mix things up, like you are being told, can you explain mitosis? So when you are told to explain mitosis, and always remember to include diagrams. So you have to put them in order. So students would have a challenge of like, which one will come first? Is it anaphase or is it metaphase? Like you are in that dilemma. Time is running out. Like this is the last question you answer in your first hand. All that. So I came up with a mnemonic from my name. So my name is Mega. Yeah, my name is Mega. And then the other name, it's Matthews. So one of my names is this, Matthews. So the mnemonic I came up with was, I called myself Professor. So this is Pro. So this color is so bad. What can we get? Okay, so the mnemonic I came up with, I said Professor Matt, Professor Matt for Matthews. Okay, so here the first stage, it's prophase. So I think you can even say I wrote these in blue on purpose. So like we have prophase, and then from Matthews, Matt, Professor Matt. This is M for metaphase, A for anaphase, and T for telophase. Yeah, so I like coming up with mnemonics. In case of anything you might forget, and all such kind of things that happen in exams. Okay, so this was supposed to be a very short lesson. Now it's just that I like talking. Okay, so we'll end here. We're done with this lesson. So we have successfully looked at the overview. We have drawn the organogram, the organizational chart. We've looked at interphase. We've explored G1, S, and G2. Right, so it was good having you. See you in lesson 2. We'll come and do mitosis. Right, so until then, bye bye and happy new year.